Welcome to the Protocol Particular series, a video interview series where we meet with members of the ASAP CRN to discuss their protocols. For today's episode, I'm Devin Snyder and I'm going to be hosting. And with me, I have Natalie Doig, who is a postdoc at the University of Oxford in the McGill Lab and is a member of Team Cragg. Hi, Natalie.
Hi, Devin. Nice to meet you.
You too. Thanks for being on today. So for today's episode, we're going to go over your collection of protocols called the quantitative analysis of the ultrastructural features of dopaminergic axon terminals. But before we go into that, I'm wondering if you could briefly tell everyone listening about your team's ASAP projects and also maybe go into how the collection fits into that project.
Yeah, sure. So this ASAP project that I'm working on kind of part is the wider research network because I'm actually not funded directly by ASAP. But what the project is really about is about looking at the striatum and the neuromodulators in the striatum and seeing how this becomes dysregulated in Parkinson's disease. So we know that a lot of things go wrong with dopamine in Parkinson's, but also looking at other neuromodulators within the striatum. But part of the wider part of that is looking at this from many different angles. So a lot of the work that we do in our lab is kind of in vivo work in mice and mouse models of Parkinson's disease. And that can be like recording from hundreds of neurons, looking at changes in dopamine and neuromodulators in behaving mice and in Parkinson's model of behaving mice. But it's also to do with the circuitry of the striatum, connectivity in the striatum, and connectivity kind of beyond the striatum within the basal ganglia. And this protocol kind of fits into that because this protocol was designed, has been in design for a long time, but the idea is that if you see fundamental changes in a disease model of Parkinson's disease, and we can see, so for example, if we see that neuromodulators change significantly in the disease, then we can look at the underlying structure of that and see how that might underpin some of these physiological changes that we see.
That makes a lot of sense. So it makes sense as to why the protocol's the ultrastructural features. So then I think we should just transition into going into some of the more specific information about the collection. So the collection is split up into two different protocols. The first focusing on tissue prep for electron microscopy. And I noticed there were four major sections, perfusion, tissue prep, immunohistochemistry, and then preparing the sections for electron microscopy. So I thought we would just go through each of those sections for all of our listeners. So as I said, the first one is on perfusion. And something that I noticed when reading the protocol is that you made a really nice note for anyone who's looking at the protocol about using the glutaraldehyde and how that's going to be essential for preserving the tissue for EM, and it should be added to the paraformaldehyde fixative. And so something I'm just not familiar too much with electron microscopy. So something I was wondering was like, if your group determined that you needed to add the glutaraldehyde, or if that was something that was already known in the field.
So I guess the use of glutaraldehyde as a fixative has been around for decades. The kind of nuance here is that glutaraldehyde is like a really great fixative, but if you use too much in terms of concentration of that, then your antibodies won't work because you lose kind of immunogenicity the higher your tissue will be beautifully fixed, but you won't be able to look at anything with an antibody. So the idea is always about trying to find that compromise between having really nice fixation of your tissue and also allowing, making sure that your antibodies can still work and you're not completely blocking the antigens that you're interested in. And this is something that you have to play around with sometimes. So for this particular antibody and tyrosine hydroxylase as a marker of dopaminergic terminals, the antibodies that we have are really, really good, and there's a range of them available out there. And so we're using a concentration of glutaraldehyde that's slightly on the higher end So if you were looking at something that maybe a different antigen you were interested in, and you weren't getting any staining with your antibody, then one of the first things that you could go and tweak would be to reduce that concentration of glutaraldehyde and just see whether that would rescue kind of the antigenicity.
That's really fantastic to know, because that was actually one of my next questions. Was I saw in your protocol, I think you used like 0.1% volume per volume.
Yeah.
And so, but you said there was a range. And so I was like, but do we need to optimize this? Does that
Yeah.
for the immunohistochemistry work for like most antibodies? Like,
Yeah.
so thank you for saying that. So then I also noticed for that section, I also
Yeah.
haven't done perfusions before, but you mentioned that you do the perfusion starting off with PBS, and then you switch to that PFA, glutaraldehyde fixative. And so I was just trying to think through like how, like physically, how your hands would actually
Yeah.
work, and like how difficult that would be, especially for someone who hasn't really done that technique very often. Like, I don't know if you have any tips on that, or maybe it's not difficult at all.
So I think in terms of the perfusion, the key thing that you want to do in the perfusion is the first step is to use your phosphate buffered saline to get rid of the blood. And you want to do that as quickly as possible. And then you want to switch to the fixative solution. And physically, how we do that is it's simply tubing that has two sort of clamps that close it. So you close one clamp and open the other, and that's how you switch. So it's not too difficult.
Gotcha.
But there are lots of yeah. There's
I
lots
was
of
like
different
picturing
setups.
multiple pipettes or something.
Yeah. Yeah, so there's
All
many
right.
different setups. Some people use pumps, some people don't use pumps, some people use syringes. Yeah, I think some people feel very strongly about how to do a perfusion in a certain way. For me, the key things is getting the blood out quickly, and getting the fixative solution into circulation as fast as possible, basically.
And so, how quick is quickly, do you think, or what would be like an optimal amount of time?
So probably I would aim for about 2 minutes of PBS, 2 to 5 minutes. Because what happens is if you leave it any longer, then enzymes start breaking down and you start getting degeneration in the brain. So you want to kind of fix everything just as quickly as you can. If that makes sense.
Yeah, that does. So then the next step in your protocol or major section is on the tissue prep.
Yeah.
And you mentioned that there's a common error with trimming unevenly. And so I was wondering if you could just go over like your tips for avoiding that and what the issues are if you do trim unevenly. Is there any way to like rectify that, or you just need to start over with a new sample?
So it's sort of so for electron microscopy, you have to use a vibrotome to cut the tissue, as opposed to what people commonly use, like a cryostat or a freezing microtome, because if you use cryostat or freezing microtome, you can damage the tissue through this freezing process. So you have to use the vibrotome for EM. And so to get a nice kind of block and even tissue, we embed that into agar, the brain into agar before cutting it. And one of these common things that you see is that, and it's especially important if you're doing an experiment where one hemisphere is your control and the other hemisphere is your experimental hemisphere, you want those two hemispheres to be, in the same plane. So if you trim it unevenly when you're cutting, then just those two different planes aren't matched up. The way to avoid that is to trim your brain initially using what's called a brain matrix, which is like this sort of mold, I guess, that is shaped like the mouse brain in this case. And you put the mouse brain into that, and it has these kind of handy little cutting marks, so you can trim it evenly. And even then, it's not always perfect, but you can get it as best as you can, basically.
Gotcha. That does sound very handy. And then you mentioned that you embed the brain in the auger. And I think I read something about obviously using cool auger, but that to make sure that the brain like doesn't float around in that.
Mhm.
So I was wondering, like, does that happen often? Does it easily start moving? And like, what do you do to try and avoid that?
Yeah, so we have some very like I don't know what the word is, like DIY, like it's basically a cut off falcon tube. And essentially I put the brain at the bottom of the falcon tube and then pour the agar onto it. And you just kind of have to pour gently, perhaps use a pair of forceps to keep it in place. And then
Gotcha.
after you've done that, you can kind of trim the agar nicely.
Okay.
But yeah.
Very cool. The DIY. I love
Yeah.
good DIY. So then the next section of the protocol talks about the immunohistochemistry
Mhm,
using the silver intensification, and that you
mhm.
do that reaction in the dark. Do you do that in like a dark room, or are you able to place the samples in like a box? How do you actually go about doing that?
So also probably a little bit DIY. I just use foil to
Oh,
cover my samples while I'm doing the reaction.
that
Um,
works.
yeah, yeah.
Cool.
We use a lot of foil in the lab, actually.
Good resource then. And then do you have anything else that you think viewers might want to know about the immunohistochemistry section that you'd like to give any recommendations
Yeah, I
on?
think, I mean, this protocol was written specifically for looking at tyrosine hydroxylase as a marker of dopamine, but there's no reason why you couldn't look at a different neurotransmitter or a different marker of terminals that you're interested in. And I guess the tricky thing, this is an antibody that we've used, it's tried and tested, the concentration works. The tricky part is really getting those concentrations right in the first place. So basically you would just, if you wanted to look at something else, you would just have to troubleshoot it from the beginning, basically, to find that concentration that works for you.
Yeah, that makes sense. And so then the last section of this protocol is about preparing your brain sections for electron microscopy. And I know there's like a lot of subsections within
Yeah.
that. I think like about like using the osmium tetroxide, there's like a dehydration process and embedding in
Yeah.
resin, and then re-embedding for the electron microscopy. So it's kind of a lot. Just so for the sake of time, I wasn't going to go into super specifics, but I was just wondering for like across that, if there were critical steps that you felt others might find to be tricky.
So I think in once you've done the staining and you're happy with your stain, then it is really just getting the tissue ready for the EM. And of course, in the electron microscope, what you're using to create contrast is this beam of electrons. So a huge part of those steps, like the osmium uranyl acetate, is just you're adding heavy metals and you're getting them to bind into the tissue, and that's what's going to create the contrast when you actually get to the electron microscope. So it's kind of, yeah, you just have to do these steps. The osmium is essential. So the osmium is absorbed by lipids. So that's what gives you your kind of your definition of your lipid membrane, and it's also absorbed by the myelin. And so that's a crucial step. And again, depending on the staining protocol that you have, you might need to titrate, and the thickness of your sections, you might need to titrate the time that you're doing your osmium staining for. And again, it's a compromise. You can do the osmium staining for longer, you get more contrast, but in the case of the silver intensified gold, you start to lose gold staining. So you
Okay.
kind of, so you have to kind of compromise between these different things. And then the dehydration is essentially you want to get all the water out of the tissue, and you want to then infiltrate the resin into the tissue. Because that is also going to be then cured and also going to add to that kind of preservation of your ultrastructure. So yeah, it's a lot of steps, but yeah, I think, yeah, if you follow the protocol, they're fairly straightforward. Although
Yeah.
there's a lot of horrible chemicals in there, so you have to be very careful.
Yeah. So safety, be
Yeah,
safe.
safety, yeah. Yeah, and I tried to make sure I included in there like safety notes for everything.
Yes, I did see that. That was really great. Okay, so then that's the end of protocol one. So then you're all ready to do the EM. And so you want to like acquire your images and actually analyze that. And so that's
Mhm,
protocol number two.
mhm.
And so I saw that you have like this really great section in the very beginning of the protocol that talks about sampling and the image acquisition. And so I just was wondering if you could briefly cover like typical mistakes that people might make when
Yeah.
sampling and why it's so important to avoid those.
Yeah, so I think over the course of using this protocol, we thought quite a lot about how to do the sampling. So um, with EM, you can just get so much detail from such a small area that you have to think about your sampling strategy carefully, because you don't want to bias yourself by, you know, taking loads of images from a very, very tiny part of the striatum in this case, because you don't know, there could be certain nuances, like you could be in a different compartment of the striatum and so forth. So it's about kind of making sure that you're getting your data from, enough of a region of the striatum that you're sampling it in a kind of unbiased way. And because every block, so called, of like the re-embedded tissue that you're looking at is really, a couple of millimeters or less, that you have to think about. You're such a small area, you don't want to get all your data from that tiny area. So it's about having enough mice to begin with, then having enough of an area of the striatum, so having a few sections in the rostrocaudal plane, and then even from within one section, not just sampling from one exact area. So, and also trying to make that as efficient as possible, right? So that you're not analyzing hundreds and hundreds and hundreds of images. So yeah.
Gotcha. That makes a lot of sense. So I guess typically, like, what's the number of images that you might take from a section?
So from one ultra thin section, probably 25 images. It really depends on what you're looking at. I mean, because in the striatum, the dopamine terminals are everywhere. It's very quick, relatively speaking, to get 25 images. If you're looking at terminals of perhaps another neurotransmitter, or using a different marker of something that's not as common as the dopamine terminal, then you might want to sample less, because it's just something that occurs less frequently. So dopamine is relatively easy to sample in the striatum because the terminals are just everywhere. So I think it depends also what you want to look at.
Gotcha.
But then, yeah, you might want, so from each animal, you might be looking at 100 images, something like that.
I see. So then I've not done this before. So something that I was wondering, when looking at like your example images, like you made it
Yeah.
super clear where like the axon terminal was, but I was wondering for like the untrained eye trying to do the image analysis, like how difficult might it be to like delineate these, different structures? And do you have any tips on
Um,
that?
yeah, so I learned how to do EM when I was doing my PhD. And I remember for the first, I don't know, week or so, I felt like I was just looking at, the moonscape. It really meant nothing to me. But then once you start to see it, it just kind of becomes very, very clear. Dopamine term, I mean, of course, in this situation, in this protocol, you would have your gold labeling. So that would be able to then, draw your eye to those terminals. And then you would be starting to look for certain features that are that you would see in an axon terminal. So the most obvious one being vesicles. If there are synaptic vesicles, then you know, okay, this is an axon terminal. Dopamine terminals are slightly different, I guess, in that they can have kind of any shape, and that the vesicles tend to kind of fill the entire terminal. They don't look like your kind of quintessential, diagram that you would see in a textbook of this is the synaptic terminal, and this is where it's forming a synapse, and this is where the vesicles are. They kind of look a little bit more sort of less defined in shape, I would say. And they make symmetric synapses rather than asymmetric synapses, which are kind of less often, less obvious, I suppose, is the word.
I see.
Yeah.
So hopefully people who are going to be doing this protocol have someone like a good mentor to help show them.
Yeah. I mean, there's also lots of published literature out there as well that could guide you. But yeah, it definitely...
The gold also.
Yeah, the gold staining will definitely help you to recognize those things.
Well, so that was all the questions I had on protocol number 2. I don't know if there was anything else that you wanted to add. If not, perfectly okay.
No, I just think that I would maybe just say that, this is a protocol that we've been working on for, I think we first published with something similar like 10 years ago. So it's kind of been in refinement over that time. And it uses a piece of software that was designed by a guy called Max Larsson, who works in Sweden. And he wrote this piece of software, and then I started using it, and it's like open source, available on GitHub. And I guess I just want to say like how important open science is, because basically I emailed him and explained what I was trying to do, and he was incredibly helpful and implemented some features into that software that I was interested in. And we've published, I guess, together several times since then. And I guess it's just kind of, when you're using this kind of open source, freely available software, how useful it is if you can talk to the person who wrote it. But yeah, I think that's it.
Yeah, that's amazing. I'm really glad that you shared that story about your science or your experience with open science. And that's part of the whole reason that ASAP asked for our members to put their protocols on protocols.io and make them open for other people to see. And so based on that, I'm assuming that you would be open to people contacting you if they have questions throughout this protocol. I think there's a little contact button on the protocol actually in protocols.io.
Absolutely. And I really believe in reproducibility. And I think that some of these, this is quite a nuanced technique, I guess. And it can make all the difference if you can reach out to somebody and say, okay, what did you mean by this? Or what does this actually look like in practice? And I also think that, going forward, the use of like video in protocols would just be so helpful. Like if I'm training students to do perfusions, for example, there's a protocol on Jove that I give to the students to look at before they do it, so they kind of know. I think if you see something visually, you have a much better idea of what you're doing. And I think and I encourage all students when they're learning, like take a photo of the setup. I think it just can make life so much easier if you can see something in a photo or a video or whatever. So, but yeah, of course, happy to help anyone.
All right. Thank you so much, Natalie.
Thank you.